court. We have 10,000 at least different molecules that we can detect. How is that possible? We're going to look today at sensory receptors. They're diverse, they're very sensitive, and they can detect an amazingly enormous array of stimuli, including all of the different smell molecules that we saw in the video. So each molecule of smell is a chemical. So the chemicals that you smell must be volatile. That means that they must be evaporated into the air, and we smell each individual molecule. So we're going to look at receptors and the marvel of them being able to detect so many different things. So then I'd like to look at general senses. General senses are those senses that are scattered throughout your entire body. Vision and chemical senses, but not necessarily in that order. Vision, I think we'll talk about next week. Vision is an enormous topic, and so it will take up an entire day. Hearing and equilibrium also will be another day. So how do you think your senses compare to the senses of other animals? So it all depends on your environment. The purpose of senses, well, what is the purpose of senses anyway? It's for us to be able to determine what our environment is and whether or not we need to respond to it. Is it safe? Is it not safe? Where is the food? For the baby, where is the food? So the first sense to be very acute for babies is the sense of smell. So that's how they recognize their mothers from other people. And interestingly, mothers can smell their babies and distinguish their baby's clothes from other mothers' babies' clothes. Yeah, it's a very acute sense of smell. So this bear, a grizzly bear, is catching salmon in a stream, has a very large rostrum. So that's a giveaway that the sense of smell is really important for the bear's survival. So the bear, once a year, somehow finds its way to this stream to get the salmon that migrate only once a year. How does it do that? Well, it leaves its own smell on pathways, on trees, and then it can find its way back to the source of food. Bears have a terrific sense of smell. What about the salmon? So this is a coho salmon. Coho salmon have nostrils, but they don't use them for breathing. They use gills for breathing. So what are the nostrils for? Well, to sense smell. And salmon migrate an enormously long way. So when they go out from the Fraser River, say from here, they travel all the way up to Alaska. They go out into the ocean, the Alaska Gyre. They find food, they get big, and they come back to the exact square meter of the little tributary to the river where they were born 4 years ago. So the salmon sense of smell is amazing too. Now, how they find their way back to the vicinity of the river is something that's being studied. Some people have studied the angle of the sun contributes to that. They're being able to find their way back. And also landmarks like coastlines and islands and things like that. But when they get close to the stream, they smell the very stream from whence they came, which is really cool. And their nostrils are devoted to that function. But they have other senses as well. Fish, they have mechanical senses. So along the side of a fish is something known as a lateral line. and it senses the water pressure. So they can tell where they are in relation to all the other fishes around them and to the substrate. Chemicals are sensed by a lot of different animals. Moths have an amazing sense for chemicals. So this moth is following the pheromones that are released by a female, and its antennae, this is a close-up of the antennae, a scanning electron microscope close-up. I have all these hairs, receptors, and they test the air for pheromones. So that's why if you ever see a butterfly or a moth flying around, they don't fly in a straight line. They don't go, oh, I'm going to go this direction and fly in a straight line. You never see butterflies flying in a straight line. No, instead, they taste the air for chemicals, pheromones in this case. And then they think, okay, this is a large concentration of pheromones here. I'm going to go in that direction. And they keep tasting the air. Oh, it's stronger over there. They go like that. So butterflies fly fairly erratically, eventually getting to their destination. And there are some senses that other animals have that we don't. So this is a viper, a snake, and it has an infrared sensor, infrared. And that is basically the detection of heat given off by prey. So they can sense that from a ways away. And we can also detect heat, of course, but we don't have special receptors to detect heat from a specific direction. Not as acute as the viper. Infrared is radiation that's given off by the body in the form of heat. And we have electrical, not electrical sense, we have sensors that can sense heat. So that's coming off of our body. And well, this is a shark. And sharks, they've had the same body plan for like 300 to 400 million years or so. They are the top predator, the top of the food chain, basically, in marine systems, and they have such good senses. Their sense of smell is remarkable. They can smell like one molecule of a tuna in like an Olympic-sized swimming pool. They have a good sense of vision, and they have something really interesting. These small pockets of oil called ampules of Lorenzi, where they can sense electrical activity of animals that are in their vicinity. So they can sense if something's breathing, even if it's hidden. And all of these senses of these animals and of us are due to the sensitivity of our receptors and the different kinds of receptors that can tell what we're sensing, what part of our body is sensing it, and we can even perceive it very specifically. So I'd like to look at receptors and their properties. So in the video, we looked at taste receptors. Taste receptors are cells, and they have hairs that are stereocilia that are receptors for different chemicals in food, whatever you taste. But not all receptors are cells. So what are receptors? What are receptors? Receptors are a structure that can detect a stimulus. So a structure that detects a stimulus. And there's so many different kinds of stimuli. There's chemical stimuli, there's light stimuli, there's so many different kinds. And the receptors can be simple or they can be very complex. So light, for example, is detected by the eyes, and that's a complex organ. So they may be complex organs that enhance and even interpret some senses before they get to your brain, such as eyes, or quite simple, such as a nerve ending. Each of our hairs, for example, has a nerve ending wrapped around it. So each hair on your body has a separate signal to your brain. But do you notice it all the time? No, of course not. You're habituated. So if you weren't habituated, you would feel the clothes that are rubbing each hair on your body. And that would be like overstimulation and your head would explode. So we don't sense that. But if you had something unusual, like a spider, you can try this. Try this with your pencil. Gently rub the hairs on your arm with your pencil. Oh yeah, you can feel every single one. And that's to alert you in case something is on your arm, so a spider or something like that. And receptors are very special in that they can transduce all of these different kinds of stimuli, light, chemicals, stretch, into an electrical signal. So they're called transducers. Receptors are transducers. They perform transduction, which is converting a signal to an electrical signal, or I should say a stimulus to an electrical signal. So they convert stimulus energy into electrochemical energy. And that is known as sensory transduction. Amazing. So what sensory transduction is, and it occurs in different ways. But here is a stimulus. Say it's light. Light is a stimulus. In the case of light, the receptor is called a photoreceptor. And specifically for light, the photoreceptors are pigments in cells. And they trigger membrane potentials. In other words, they cause the plasma membrane to be more permeable to ions. And that, in turn, causes the receptor to release neurotransmitters which, guess what happens then? Triggers potentially an action potential in a first order sensory neuron. So that, my friends, is transduction. Transduction. Okay, well, then how does your brain know if it's blue light, red light, green light, magenta, burgundy, lilac? I mean, we're detecting a lot of different colors. We're detecting a lot of different colors. How does our brain know that it's light and not a chemical that we're detecting? How does the brain know how intense that chemical is? Is it a chili pepper, like super strong? Or is it mild, like porridge? So the information conveyed to the brain, so the information about a stimulus, what is it? How strong is it? Conveyed by sensory receptors are the type of stimulus, also known as modality. What is it? Is it light or something else? So neurons from the eye, if the neuron is coming from the eye, that alerts the brain that it is light. Interpreted as light. What else can the brain discover from the sensory neuron? Well, the location of the body from which that stimulus is coming from. So each receptor, each sensory receptor, receives input from what's known as a receptor field. From a receptor field. So location of the body. I'll show you a receptor field in a moment. The receptor can also convey how intense that is. And that is delivered via the frequency of action potentials. And also the number of neurons that fire. So there can be many involved. We also get information about the duration of the stimulus. So some receptors are known as phasic receptors. Phasic receptors. They get bursts of activity and they adapt very quickly. So this is just some terminology for different kinds of receptors. Phasic receptors. Burst of activity and quickly adapt. So we've talked about one of those, the receptors on your hairs. And interestingly, smell receptors adapt very quickly too. So if you smell something bad, like a skunk or something, if you're in the vicinity for a while, eventually you'll just adapt, and you won't be consciously smelling that smell for a very long time. Tonic receptors are those that adapt slowly. Um, because you are in need of that information. So proprioceptors, the ones that are on your muscles and your tendons, or in your muscles and your tendons, that tell your body what position it's in, those adapt more slowly because the body always has to be aware of the position of your body. Sorry, the brain has to be aware of the position of your body all the time to adjust for movement so you don't fall over. But all receptors will eventually adapt. So conscious sensations often decline over time. If there is no purpose, no survival purpose, or no balance purpose to continue responding to that signal. So here's a receptive field for touch. So this is a single neuron, and it has a broad receptive field. And these are 3 separate neurons that each have their own receptive field. So we're going to test this. Well, you're going to have to test it on yourself, I think, for the Senses lab. So which part of your body is more sensitive, do you think? The tips of your fingers or your arm? So for example, if you had 2 stimuli like this, and they were this far apart, if you touch them with your eyes closed, somebody else would have to do this, of course. If you touch them on your finger, you would be able to tell there were 2. Yeah, good, your fingertips, yeah. If you were to touch them up here with your eyes closed, you would just think they were 1. Excellent. So how can we classify receptors? I would really like to have lined paper here, ruled. There we go, there. So a classification of receptors. Classification of receptors. So this is just a way of identifying the receptors themselves. A modality or type. How many types are there? I'm just going to write on here. There are chemoreceptors. So where do you think chemoreceptors would be important? Where would chemoreceptors be important? Which senses? They sense chemicals. Where are chemoreceptors in your body? Your nose, good, yeah. So your sense of smell, your sense of taste, and the composition of body fluids. Good. Modality. Thermoreceptors. That detect the mucous membrane. Good, yeah. For chemoreceptors, excellent, yeah. Thermoreceptors, we have both external and internal thermoreceptors. We have nociceptors. They detect pain, damage, poor blood flow, excessive heat, excessive chemicals. We also have two other ones, mechanoreceptors. That detect such things as stretch and photoreceptors. That detect light. So that's classification by modality. We can also classify them by origin. So some are called interoceptors. They detect internal visceral stimuli. Pain, nausea, stretch, pressure. Proprioceptors is another classification. And they're found in joints and muscles. They detect muscle and joint movement. And last but not least are the external ones, exteroceptors. So they detect light, for example, smell. So external stimuli, such as light. That's just a way of classifying them. We also can look at them by distribution. What is their distribution in the body? So general senses are somatic senses. And they're widely distributed in the body. Widely distributed. Touch, for example, pressure, heat, cold, pain, blood pressure. And blood composition of chemicals. So those are your general senses that are distributed widely in the body. The other ones are special, and they're not distributed widely. They're just in your head. Yeah, limited to the head. And they're innervated by cranial nerves, vision, smell, taste, equilibrium, balance, and hearing. Classification of receptors. So let's look at the general senses first. The general senses. They have some remarkable receptors, really remarkable. So some are unencapsulated. That means that they're not covered in connective tissue. They're not covered in connective tissue. They're, for example, free nerve endings. Warm receptors, cold receptors, and pain receptors are in epithelial tissue found in epithelial tissue. Hang on, that's not fitting there. Free nerve endings found in epithelial tissue. And connective tissue. It's the most abundant. Tactile discs. Tactile discs. They're also free endings. They're not called free nerve endings, but they're unencapsulated, but they're formed in a disc. So they're concentrated. They're a concentrated nerve ending. So they are better at detecting subtle things like light touch, for example. Light touch, texture, the texture of objects, the edges of objects, and shapes. And tactile discs are found, as you can see here, in the base of epidermal tissue. Quite sensitive. Hair receptors monitor the movement of hairs. Yeah, and of course they're found on every hair, wrapped around the base and the follicle. Yeah. So they're not all unencapsulated. Some of them are encapsulated, which makes them more sensitive. It enhances the sensitivity of that receptor, which are all nerve endings. So they're known as tactile corpuscles, Krauss end bulbs, lamellated corpuscles, and Ruffini corpuscles. And so where you find them is in different parts of the body. So for example, tactile corpuscles you will find in your fingertips, very sensitive. The palms of your hands, you'll find that your palms are quite sensitive. Your eyelids, very sensitive to protect from debris getting into your eyes. Lips are also very sensitive. Nipples, very sensitive. And genitals. So that's where you'll find tactile corpuscles. So the tactile corpuscles detect light touch and texture in those locations. The Krauss end bulbs you find in mucous membranes. Lamellated corpuscles, basically what that is like a Schwann cell that's wrapped the end of the nerve ending. So the lamellated corpuscles will detect, what kinds of things will they detect? Deep pressure, for example, stretch, tickle, itch. And you find them quite deep in the dermis, particularly in the hands and feet. Ruffini corpuscles, they detect heavy touch. You find them in ligaments, tendons, and joints. And the Ruffini ones, detect pressure, joint movements, skin stretching, pressure. So those are unencapsulated and encapsulated nerve endings for senses such as heat, cold, touch, pressure. So how does that information get to the brain? Through projection pathways. The first order neurons from below the head, to distinguish them from cranial, enter the dorsal horn of the spinal cord. From the head, they enter the pons or medulla. And they're not all the same. So the first order sensory neurons are not all the same. Some of them are small and unmyelinated, and some of them are fast and myelinated. So fast myelinated, and they carry signals such as proprioception. Some of them are unmyelinated and smaller and slower. So they carry signals such as heat and cold. So you don't get as fast a signal from heat and cold. The second order neuron synapses with the first order neuron in the spinal cord or the pons or the medulla and crosses over to the other side, which is decussation. So decussation occurs to opposite sides of either the spinal cord, depending on which one it is, or the medulla or pons. I guess the medulla mostly. Yeah. And they end in the thalamus. Except for the proprioception ones, they end in the cerebellum. Proprioception, second order neurons end in the cerebellum. And the third order neuron, which synapses with the second order neuron in the thalamus, projects to the primary somatic cortex. So they extend from the thalamus to the primary somatic cortex of the cerebrum on the contralateral side, so the opposite side. So it's interesting. There was a study done by, well, this is quite a long time ago, a psychologist or a psychiatrist, I guess, who noticed that in like portrait pictures of women with babies that they tended to hold the babies in their left arm. And he thought that was very interesting. So it's about 90% or so. And he wondered why that was. So there were some hypotheses. One was that the head would be close to the heart. But the head isn't really close to the heart there. It's over here and the heart is here. So that was not likely anyway. And he thought, well, maybe it was because if they're held in the left arm, then the right arm, because most people are right-armed, or right-handed rather, would be available to do work. But then he discovered that even left-handed people would hold the baby in the left arm. So yeah, but that was not really that likely either. But then I think the final hypothesis, whether it's actually been shown to be the case or not, but it was interesting, was that the right side of the brain tends to interpret things like facial expression. And of course, babies have lots of facial expressions that indicate, that they're in distress. So it's thought that by looking on the left arm at a baby, that would be received by the right side of the brain and interpreted more readily. Yeah. The third order neurons ends the somatic contralateral. Compared to the first order neuron. Yeah. Yeah, that's a good question. So what about pain? Yay. Well, let me ask you this question before we look at that. Let me ask you this question. Why do we feel pain? Why do we feel pain? To stop doing what we're doing that damages us. Yeah. So it brings attention to an area of damage so that it doesn't get further damage or infected. So in conditions like leprosy and diabetes mellitus, the sense of pain can be lost. Due to nerve damage. Nerve damage is collectively known as neuropathy. Pathy means disease or disorder. Neuropathy. Yeah, so pain makes one aware of injuries. It makes one aware of injuries to the body. So they can be treated to avoid further injury and infection. So the question is pain connected to pleasure. Well, what pain can do is release endorphins. And endorphins, I'm not sure exactly which one, but we are going to get to that. They can block pain signals and they give you a feeling of euphoria. So that's the connection there. So pain is a very necessary adaptive kind of sense. The name of the receptors is a nociceptor. It makes you conscious of pain. It's found in all tissues of the body except the brain. The brain doesn't have nociceptors. Fast pain travels in myelinated fibers very quickly, 30 meters a second. That would be like a sharp, localized pain. That would be a fast travel, a stabbing kind of pain. But slow pain travels more slowly, but it's diffuse. So you have a longer lasting. It's longer lasting than sharp pain. And diffuse, so more of a dull kind of feeling. Somatic pain comes from the skin, the muscles, and joints. Visceral pain from stretch, chemical irritants, or ischemia of the viscera. So that is a lack of blood to the area. If tissue is injured, it releases chemicals. And it's those chemicals that stimulate the pain fibers, the nociceptors. And the chemicals are known as bradykinin, histamine, and prostaglandin. So those are chemicals released by injured tissue. So the general projection pathways for pain. So the first order neuron in the dorsal root ganglion of spinal nerves except for the trigeminal nerve. It has its own ganglion. And that transmits pain from the face. It's the trigeminal nerve that is dulled when you go to the dentist, because that's the nerve in the mouth that feels pain and that even detects pain, or that detects really spicy food like chilies, habaneros, and stuff like that. Yeah, so the spinal-reticular tract carries the second order neuron. which also decussate. And end up in the thalamus as well. Where they synapse with the third order ones, which also reach the primary somatic cortex. Third order to somaesthetic. If you write somaesthetic enough times, you'll remember it. The somaesthetic cortex. That's my plan. My evil plan for you to write things a lot so that you can remember them. A referred pain is misinterpreted pain. So the pain, the brain assumes pain is coming from the skin or some other superficial site. And that's because some signals from the skin follow the same pathway as signals from some of the viscera. So the brain misinterprets. So heart pain, for example, is felt in the shoulder and arm. Because they both send pain to the spinal cord segments T1 to T5. Thoracic vertebra 1 to thoracic vertebra 5, part of the spinal cord. So because of the location of the signal, the brain can misinterpret. So let's see what that looks like. So here is a nociceptor in the finger. Youch. The first order neuron travels. There's the soma to the dorsal horn and synapses with the second order. The second order neuron, which goes over to the other side, occasionally will go straight to the thalamus, where it synapses with the third order neuron, which continues on to the primary somatic cortex. Occasionally, some signals will go through what's known as the reticular formation, which will do some integration of signals and then send that signal to the primary aesthetic cortex. So those are the pathways of pain. And let me show you some referred pain areas. So this is how a medical practitioner may be able to determine which viscera are affected by where the pain is in the body. So you don't have to memorize the locations and parts of every single one of these, but do be aware of what referred pain is, and just remember the heart example. But here you can see the lung and diaphragm, for example, might be detected in this part of the body, the heart, shoulder, arm. Yeah, referred pain. So does the central nervous system have anything to do with pain? Does it modulate it? Yes. Yes, it does. For example, it can be affected by the state of mind. If you are in a stressed or complex kind of state of mind, then you're likely to feel pain more readily. So the central nervous system, it does have pain relieving mechanisms. So the mechanisms were discovered through, interestingly, the actions of opiates, so morphine and heroin. So they discovered receptor sites for those particular drugs in the brain. And as it turns out, we have drugs that bind naturally to those sites. They're called endogenous opioids. Enkephalins, endorphins, and dynorphins. So these are produced by the central nervous system under times of stress. So stress occurs when you're in pain or exercise. So exercise does cause some pain, some muscle pain, some joint pain, although it's not that detectable by you. Your body interprets that as maybe potential pain and releases endorphins.
Maria, we can't see the updated slide, I think.
Oh, okay. Let me do this. Mm-hmm. We had this problem before. I wish I could remember how we fixed it. Let's see. There. Oh, so you stopped at this one. You couldn't see that one. Is that right? It's okay now. Okay, good. Yeah, so this is the referred pain slide. Sorry about that. I did not notice. Concentrating pretty hard here. So yeah, referred pain. The lung and diaphragm, for example, might be detected in the neck. And modulation of pain. So the intensity is affected by the state of mind. Endogenous or opioids that are produced by you are enkephalins, endorphins, dynorphins. They're produced by the central nervous system. Stress or exercise. And they're found especially in the dorsal horn of the spinal cord. And that explains spinal gating, which stops pain signals. So they also act as neuromodulators. The endogenous opioids block transmission of pain and produce feelings of euphoria, so I don't know if you have you ever done really extreme exercise, or even you don't even have to do very extreme exercise. You can even like walk fast, and you'll release endorphins, which will block pain and make you feel really good. That's what's thought to cause, the runner's high. So when people run for a long distance and then they feel like really euphoric, and that's why they feel super euphoric. Secretion rises very sharply in women giving birth, for example. So that is really all I wanted to say for now. I'm going to stop there. Thank you for watching.